Assalamu alaikum. This is Dr. Mamoun Ahram. I'll be giving you the second lecture on this module. We'll be talking about the biochemistry of neurotransmitters. If you have any questions or feedback, if you have any problem, please send me an email. If you like it, you can send me an email as well. So good luck. Thank you. So let's start with the biochemistry of neurotransmitters. These are the resources. The lecture is the main source for information. You can refer back to Mark's Basic Medical Biochemistry. You have the pages here. It's about 10 pages. Or there's this nice website that combines biochemistry with anatomy. You can refer to it as well. It has nice and simple information on this topic as well. So let's define neurotransmitters first. A neurotransmitter is defined as a chemical substance. That is, it has to be synthesized in a neuron, then it is released in the synapse following depolarization of the nerve terminal, and then it binds to a receptor on the postsynaptic cell, inducing a signal transduction on that cell. Some of these neurotransmitters can bind to receptors on the presynaptic terminal as well. Eventually, a specific response is elicited. You have a certain induction of certain response, whether it is muscle contraction or something else. All right. So these are the characteristics again, or the condition for a molecule to be a neurotransmitter. It has to be synthesized and stored in presynaptic neurons. The enzymes needed for synthesis must be present in this neuron. The chemical substance is then again released in the synapse following depolarization. It binds to a receptor, and then it elicits a rapid onset, and this onset is reversible as well. And it has to be removed or inactivated from the synaptic cleft so that the signal is terminated. Now, there are different types of neurotransmitters. First, you have small molecule neurotransmitters, and these can be divided into different subclasses as well. So sometimes you can have biogenic amines like epinephrine, dopamine, histamine, serotonin. You can have amino acids, surprisingly. You can have amino acids or modified amino acids as neurotransmitters like glutamate, aspartate, glycine, or you can have GABA. We'll be talking about GABA at the end of this lecture. Acetylcholine and ATP are also small molecule neurotransmitters. You can have large neurotransmitters known as neuropeptides, or you can have really small and short-lived ones known as gases like nitric oxide as well as carbon monoxide. Sometimes a neuron may have more than a neurotransmitter. They can have a combination of small molecule neurotransmitter or a neuropeptide in the same neuron. So these are the different structures for the molecules. You have the biogenic amines, really small. You have the ATP in the bottom, and to the right you can have the neuropeptides that are composed of very short sequence of amino acids combined to each other. So let's start with neuropeptides. Now, there are many neuropeptides identified so far. They are responsible for different behaviors or different responses like pain perception, memory, appetite, thirst, and so on. There are many of them, and not all of them have been discovered so far. Now, is a neuropeptide a neurohormone or a neurotransmitter? There is a distinction between the two. The same molecule can act as a hormone or it can act as a neurotransmitter. It depends on where it acts. So if it is released from a certain cell and then it travels, let's say, in the blood or the lymph, and it travels a long distance, then it is known as a neurohormone. Otherwise, if it functions within the same region where it is released, then it is known as a neurotransmitter. So they can be classified into different types according to structure or function. You can see that you have different neuropeptide families. For example, you have the insulins, you have the somatostatins, gastrins, and so on, and each one of them can contain different types of neuropeptides. You have the opiate family as well. They have similar structures if you look at them. They have the same the 4 amino acids in their structure, but they differ in the amino acids that come afterwards. So you can look at the leu encapsulins or the met encapsulins. depending on the presence of leucine or methionine in their sequence. Now, you have two neuropeptides like vasopressin and oxytocin. They share 7 out of 9 amino acids, but they have totally different functions. And these two different amino acids, regardless of what they are, they give this distinct function for each one of them. Now, I talked about the opiate peptides. They share a common sequence, but they bind to different receptors. And we have also glycoprotein hormones like the thyroid stimulating hormone or the luteinizing hormone and the follicle stimulating hormone. They have the same alpha subunit, but they have a different beta subunit, and that's what gives them their certain structure and function as well. Now, so let's talk about the synthesis of these neuropeptides. They are synthesized starting in the endoplasmic reticulum and the Golgi apparatus, with the rough endoplasmic reticulum actually, and these neuropeptides are inserted into the endoplasmic reticulum where they get modified. Then they are transported into the Golgi. They are modified further and packaged into vesicles known as large dense core vesicles. They can also coexist with modifying enzymes in these vesicles, and during their travel from the cell body to the terminus of the neuron, they can be modified further by these enzymes. They are transported via fast-axonal transport along the microtubules all the way from the cell body to the cell terminus, where they wait for a signal to be induced, and this signal elicits the opening of channels, the entry or the influx of calcium ions, and that induces the fusion of these vesicles with the plasma membrane, and the neuropeptides are released. Their action is prolonged. They are stable for a long time, and they get terminated by either diffusion or degradation. So you can have enzymes outside the cell that degrade these neuropeptides. Again, they are real stable, and they can travel a long distance in blood or the lymph. There's diversity of these neuropeptides, and this diversity can stem from alternative splicing. So here's an example of substance P and other neuropeptides like neurokinin A, for example. They can all be produced from the same gene, except that you can have alternative splicing resulting in the production of different neuropeptides. So you can also have diversity as a result of proteolytic processing, whereby you can have one polypeptide that can be degraded in different manners, generating different types of neuropeptides. And this is determined by not only proteases, but also the packaging of the different proteases in different vesicles. They can be produced where you can have the proteolytic site hidden, and this can be controlled by cell, and that's why the production of neuropeptides is tissue specific depending on how they are regulated. An example is the pro-oplomelanccortin precursor, where it can be degraded into different types of neuropeptides like the adrenocorticotropic hormone or the corticotropin like intermediate lobe peptide, and so on. So different types of neuropeptides can be generated from the same polypeptide. There are different levels of regulation of these neuropeptides. It all starts with the type of stimulus, as well as induction of transcription and translation of the neuropeptides, starting from the gene level, as well as processing in the endoplasmic reticulum, as well as the Golgi apparatus. You can have different type of glycoproteins attached to the neuropeptide. Now, it all depends as well on the presence of proteases in the vesicles, where processing of the degradation or proteolytic processing of the neuropeptide takes place. Eventually they get released, and this can be regulated as well. So calcium ions, as I said before, are important for inducing the release of the fusion of the vesicles with the plasma membrane and the release of the neuropeptides. There is an important factor here that calcium plays, or an important points that I have to mention about the influx of calcium ions. The first one is that little amount of calcium is needed to induce the fusion of vesicles with the plasma membrane and induce the release of these neuropeptides. That's one. The source of calcium can be extracellular as well as intracellular. The other thing that is important is that the entry or the site of entry of these calcium ions can be far away from where the vesicles are. And this is different than the small molecule neurotransmitters that we will be talking about later on. So let's talk about the second type of these molecules, the small molecule neurotransmitters. As I said, there are different types. We have the biogenic amines, we have modified amino acids, and these modified amino acids can be derived from intermediates of glycolysis and Krebs cycle. So let's talk about their synthesis. Now, note the differences between synthesis of neuropeptides and synthesis of the small molecule neurotransmitters. What's important here is that we're not talking about the synthesis of the neurotransmitter, rather the synthesizing enzymes takes place in the cytosol or in the endoplasmic reticulum and the Golgi. Then they are packaged into large dense core vesicles, just like the neuropeptides. The enzymes are transported along the axon to the terminus of the neuron, and the synthesis of the small molecule neurotransmitters takes place in the synapse, where the synthesis can take place in the vesicle or in the cytosol depending on the neurotransmitter, and we'll be talking about that as well. Then you have a signal that is transmitted, and that induces fusion of the vesicles with the plasma membrane and release of these neurotransmitters outside of the cell. Their action is short relative to the neuropeptides, as we talked about before, and their action is terminated by different mechanisms, including diffusion, or they can be taken up again by the presynaptic cell, or they can be enzymatically activated. So we talked about the role of calcium influx in terms of release of the neuropeptides. Now the same thing happens with the small molecule neurotransmitters, except that an important factor is that higher amount of calcium ions is needed to induce release of the small molecule neurotransmitters. That's one. And the site of vesicle fusion with the plasma membrane must be near where the calcium influx takes place. These are electron microscopic images of such vesicles fusing with the plasma membrane. You can see the sequence of events that takes place. Now these vesicles, the synaptic vesicles, are really small relative to the large dense core vesicles that contain the neuropeptides or the enzymes responsible for transporting the enzymes from the cell body to the synapse or to the terminus of the neuron. And a lot of vesicles exist near the plasma membrane, except that they cannot fuse due to the absence of the calcium. But once calcium goes in, that induces fusion of vesicles with the plasma membrane. Now, the reason behind the role of calcium, the function of calcium in fusing or fusion of the vesicles with the plasma membrane, is that you can have these proteins, particularly the synaptotagmin. They exist on the vesicles, and this protein requires calcium ions in order to be able to interact with proteins that exist on the plasma membrane. So once calcium influx takes place, calcium binds to this protein, the synaptotagmin, and that induces or stimulates the interaction between the synaptotagmin with the proteins that exist on the plasma membrane, getting the vesicle closer to the plasma membrane and inducing the fusion of the plasma membrane of the vesicle with the plasma membrane, so the vesicle becomes part of the plasma membrane, releasing the small molecule neurotransmitter. You can refer to this website in order to see an animation, or in order to see better, more detailed images of this process. Now, let's talk about specific type of small molecule neurotransmitters and how they're synthesized, because that's what's important. You would notice that there are different mechanisms of synthesizing these neurotransmitters. We'll start with the tyrosine-derived neurotransmitters, and there are three of them. We have dopamine, norepinephrine, and epinephrine, and they are synthesized sequentially. I want you to pay attention to the role of cofactors in the synthesis of these neurotransmitters. Specifically, we'll be talking about the S-adenosylmethionine, which is important for methyl transfer. We'll be talking about pyridoxal phosphate, which is derived from vitamin B6, if you remember from basic biochemistry, year 1. And this cofactor is important for transamination and decarboxylation reactions. And there is the important role of a cofactor known as tetrahydrobiopterin in the synthesis of these neurotransmitters. So this is the sequence of synthesizing the three types of neurotransmitters, starting with tyrosine. Tyrosine can be converted to DOPA via an enzyme known as tyrosine hydroxylase. This requires tetrahydrobiopterin as a cofactor. Now, of course, the source of tyrosine is either diet or liver can also synthesize this amino acid. DOPA is then converted to dopamine via decarboxylation reaction. The enzyme is known as DOPA decarboxylase, which requires vitamin B6. Dopamine can then be converted to norepinephrine, and this occurs in the vesicle, and then norepinephrine can be converted to epinephrine, and we have the requirement for S-adenosylmethionine as well as vitamin B12 or folate in the synthesis of epinephrine. This is via a transfer of a methyl group to that to norepinephrine at the end of the epinephrine molecule. Now, the first reaction, conversion of tyrosine to DOPA, is the rate-limiting step. It's the highly regulated step in the synthesis of these neurotransmitters. The other thing is that we have this moiety right here. The ring structure is known as catechol. This is a catechol moiety, and that's why these neurotransmitters are known as catechol amines collectively. The synthesis of norepinephrine from epinephrine is actually vesicular, and let's go into more details about that. So you have the transport of enzymes from the cell body to the terminus of the neuron. Right there you have the synthesis of dopamine from tyrosine. Then dopamine can be transported into vesicles, and it awaits the influx of calcium to fuse with the plasma membrane so it can be secreted. 50% of the released dopamine can be taken up again by the presynaptic neuron, whereas 10% can be taken up by the postsynaptic neuron, and the rest is actually degraded or converted or removed by enzymatically by monoamine oxidase as well as a methyl transferase enzyme. Now, what happens with cocaine and amphetamine is that they prevent the uptake of dopamine so that it stays for a longer time in the synapse, and that's why that what gives the stimulation of the neurons for a longer time, and the type of excitement or the feeling of excitement and awakening by a person taking cocaine or amphetamines. Now, in terms of the synthesis of norepinephrine, what happens is that dopamine goes into the vesicles, and these vesicles can fuse with others that contain the required enzymes for the synthesis of norepinephrine. Synthesis of norepinephrine takes place again vesicularly, and then these vesicles, which are large, dense core vesicles, can fuse with the plasma membrane, and you have the release of the norepinephrine. Again, norepinephrine can be taken up by the presynaptic cell, or it can be taken up by the postsynaptic cell. Predominantly, it's taken up by the presynaptic neuron, or it can be removed enzymatically, monoamine oxidase or the methyltransferase enzyme. Amphetamine also has an effect on the transporter that is responsible for taking up norepinephrine back into the presynaptic cell, and again you have this same effect of excitement. Now, in terms of synthesis of epinephrine, norepinephrine leaks out of the large dense core vesicles, and it can be synthesized, then it is converted in the cytosol via the methyltransferase enzyme into epinephrine, and epinephrine can be packaged into large dense core vesicles again, and again same thing, calcium influx, fusion with the plasma membrane, exocytosis of epinephrine. Most of the epinephrine is taken up by the presynaptic cell, some of it is taken up by the postsynaptic cell, or it can be enzymatically inactivated by the monoamine oxidase or the methyltransferase enzyme. So note the differences in synthesis of dopamine, epinephrine, and norepinephrine in terms of the enzymes required, type of reaction, the cofactors that are needed for each one of them, for synthesizing each one of them, the type of inactivation of the neurotransmitters, and where they are synthesized, whether synthesis occurs inside vesicles or it occurs in the cytosol. Now, how are these neurotransmitters, dopamine and epinephrine specifically, how are they packaged into vesicles? What's needed actually is that there is an ATP dependent process linked to a proton pump. So what happens is that you have entry of protons into the vesicles, and these protons can then be pumped out of the vesicle in exchange of the neurotransmitter of the dopamine or the epinephrine. So you have an ATP dependent process, and you have the participation of two types of transporters. You have one for the protons, and you have another one for the neurotransmitter itself. The name of the neurotransmitter responsible for transporting the catecholamines into the vesicles is known as vesicular monoamine transporter, or VMAT. Inactivation of the catecholamines takes place enzymatically, or as I said, diffusion, but mainly it's enzymatically. By two enzymes, you have the monoamine oxidase, or you have the methyltransferase, the catechol-O methyltransferase, COMT, and this happens sequentially. So you can have first the action of the monoamine oxidase followed by the methyltransferase, or you can have the action of the methyltransferase followed by the monoamine oxidase. Eventually, you have the formation of the final product, which is known as the homovinylic acid. A decrease in the level of homovinylic acid is associated with Parkinson's disease. So that's an interesting point as well. The synthesis of these catecholamines is regulated in the first step by the tyrosine hydroxylase enzyme. Remember, I said that this is the rate-limiting step. If you remember, the meaning of rate-limiting step is that this is a slow step in the metabolism, because it requires an enzyme that is highly regulated, and usually energy is associated with such reaction, but not necessarily true. So, but the enzyme is highly regulated. Now, this tyrosine hydroxylase can be regulated at two levels. You have the short-term level, or you have a long-term level. In terms of the long-term level, well, it takes time, because we're talking about genetic regulation, synthesis of the enzyme, or you can have the short-term regulation. It can be inhibited by free cytosolic catecholamines, so The larger concentration the catecholamines exists in neurons, the less, or the more inhibition there would be for this enzyme. Or if there is a lot of depolarization, a lot of activation of the receptor, that can also cause increase in the that can cause decrease, in the synthesis of tyrosine, in the activity of the tyrosine hydroxylase. Okay, so let's talk about another type of neurotransmitter. We'll talk about two of them, serotonin and melatonin, and both of them are derived sequentially from tryptophan. So again, what happens is that you have synthesis of serotonin, which is also known as 5-hydroxytryptamine, from tryptophan. This reaction requires tetrahydrobiopterin in the first step, that is the hydroxylation of tryptophan, and then it is packaged into the small synaptic vesicles, which again await calcium influx to fuse with the plasma membrane. Once it is released, it can be taken up again by the presynaptic cell neuron, or it can be taken up by the postsynaptic neuron, but mainly it's the first thing, taken up by the presynaptic neuron, or it can diffuse away, where it can be inactivated enzymatically via the monoamine oxidase, the same enzyme, and it can be converted into another product that can be released into the urine. So it is it's a marker for the inactivation process of the serotonin. This marker is known as the 5-hydroxy-indole acetic acid. Now, serotonin is the neurotransmitter responsible for happiness, and so its presence in the synapse can increase the feeling of comfort and feeling of happiness, and the transporter that is responsible for taking up serotonin into the presynaptic cell can be targeted by different type of inhibitors, known as SSRIs or serotonin reuptake inhibitors, like Prozac, for example. So the serotonin stays for a longer time in the synaptic cleft, and that causes again the feeling of happiness or comfort. Melatonin can be synthesized from serotonin via a methyl transferase, via an acetyl transferase enzyme. Again, melatonin is the neurotransmitter that is responsible for controlling sleep patterns or the circadian rhythm of the seasons or the dark and light cycle. Serotonin, by the way, is packaged into vesicles in a similar manner as the catecholamines. So we have two amino acids that can act as neurotransmitters. One of them is glutamate and the other is aspartate. Now, both of them are non-essential amino acids. They cannot cross the blood-brain barrier, and that's why they must be synthesized in the neurons. And there are two sources of these amino acids. One is neurons or the glial cells, and both are excitatory neurotransmitters. So, glutamate can be synthesized from different sources actually. It can be synthesized from intermediates of Krebs cycle, or from glutamine, or from aspartate. Now, in terms of the glycolytic intermediates of the Krebs cycle, actually, it can be synthesized from alpha-ketoglutarate. So here we have the different mechanisms of synthesizing glutamate. So alpha-ketoglutarate can be transaminated. It can be converted to glutamate, or glutamine can also be converted into glutamate via removal of one amino group from the outer group. Or the other thing is that aspartate can also be a precursor for synthesizing glutamate, also via a transamination reaction. Once released, glutamate can be taken up by the neuron itself, or it can be taken up by glial cells. In the neuron, once it's taken up again, it can be packaged into vesicles, If it's taken up by glial cells, and it can be taken up by different transporters, it's converted to glutamine, and glutamine is released from the glial cells all the way to the neuron, where it can be converted again to glutamate, or it can be converted to aspartate, and aspartate can be converted to glutamate. So there are different types or different sources of glutamate. Aspartate, on the other hand, is synthesized from oxaloacetate, also via a transamination reaction. Note the involvement of alpha-ketoglutarate and glutamate. Alpha-ketoglutarate is the precursor of glutamate once it's transaminated. Aspartate is converted to oxaloacetate if it is deaminated, if the amino group is removed. Now, the vesicular uptake mechanism for aspartate has not been shown, and there is still some controversy whether aspartate is a neurotransmitter or not. Glycine can also act as a neurotransmitter as well. It is an inhibitory neurotransmitter. It is synthesized from serine via 3-phosphoglycerate. So you have the involvement of the glycolysis or the glycolytic pathway in the synthesis of glycine. Synthesis of glycine requires folic acid in that process, and it can be removed via a high affinity transporter. Now, we have another amino acid or modified amino acid, which is known as the GABA, the gamma aminobutyric acid. It can act as a neurotransmitter, and it is present actually in high concentrations in many brain regions. And its synthesis, it is an important neurotransmitter that must be preserved. So there is a pathway or a process known as the GABA shunt that preserves and prevents the removal or elimination of the GABA to keep it at high levels. This GABA shunt is basically whereby glutamine is converted to glutamate by glutaminase. We talked about that before, and this can take place in the neuron itself. Glutamate is decarboxylated, forming the GABA via a decarboxylase enzyme, and this enzyme requires vitamin B6. GABA can then be stored into vesicles until released in a similar mechanism as all of the other small molecule neurotransmitters. Now, once released, GABA is taken up into presynaptic cells and repackaged, or it can go into the GABA shunt, where it's taken up by the glial cells, and this is the GABA shunt. So the GABA goes into the glial cells, and in glial cells it's converted to glutamate. Glutamate is converted to glutamine. Glutamine is released from the glial cells. It's taken up by the neuron. Again, it's converted to glutamate, same process as before. So the idea of a GABA shunt is basically to preserve GABA as much as possible, and this takes place in the glial cells. There's another neurotransmitter known as acetylcholine, a very important one, and it is synthesized from the combination or integration of choline as well as acetyl coenzyme A by an enzyme known as acetyltransferase. This takes place in the cytoplasm. There are different sources for choline. It can either be derived from diet, or it can be derived from the phospholipids that exist in the plasma membrane. Eventually, the acetylcholine is packaged into synaptic vesicles, and once released, it can be removed by acetylcholinesterase. We talked about acetylcholinesterase before in the basic biochemistry when we talked about enzyme inhibition and regulation of enzyme inhibition, and the role of the sarin gas that is used in war. Histamine is another neurotransmitter. It does not penetrate the blood-brain barrier. It has to be synthesized in the brain. It can be inactivated by two enzymes, either the histamine methyltransferase or the diamine oxidase or histaminase. Now, there's an important property of histamine is that once released, there is no way or no mechanism of taking up histamine back into the presynaptic neuron. Now, synthesis of histamine from histidine is catalyzed by a decarboxylase enzyme, and that requires pyridoxal phosphate or vitamin B6. Now, histamine is packaged into vesicles via the same transporter as the catecholamines and the serotonin, and it can be removed. Once released, it can be inactivated by monoamine oxidase that exists in astrocytes, neighboring cells. Finally, we'll be talking about nitric oxide very briefly. Nitric oxide is a gas. The synthesis of nitric oxide takes place actually in the postsynaptic nerve. What happens is that glutamate is released. It acts on receptors in the postsynaptic cell. Calcium enters. There's calcium influx in the postsynaptic neuron, activating nitric oxide synthase that forms nitric oxide in the postsynaptic nerve. Nitric oxide is generated actually from arginine. Nitric oxide is released, can stimulate guanylate cyclase enzyme forming cyclic GMP from GTP that results in a certain physiological response. Now, nitric oxide can diffuse out of the postsynaptic neuron to the presynaptic cell, inducing a certain action. This is known as a retrograde messenger. It can also diffuse into adjacent neurons as well, or like glial cells, other neurons or glial cells, stimulating guanylate cyclase in those neighboring cells. Now, an important aspect of nitric oxide is that its half-life is very short. It's about 2 to 4 seconds. And it can be inhibited by hemoglobin or other heme-containing protein where it binds to heme. There is controversy whether nitric oxide is a neurotransmitter or not. So it is a neurotransmitter, but it's different than the classical neurotransmitters that we talked about before because it's not stored in vesicles. It is not synthesized in the presynaptic neuron. Rather, it is synthesized in the postsynaptic neuron. Its release is independent of calcium influx. Its inactivation is passive. It's only by diffusion. It decays spontaneously. It has a very short half-life. It does not interact with a receptor on the target cell. Rather, it interacts with enzymes in the cytosol of the postsynaptic cell or in the neighboring cells once it diffuses out. So it depends. Its sphere of action depends on where it diffuses and the reason by which where it diffuses. Nitric oxide does not. It acts as a retrograde messenger where it goes back to the presynaptic neuron, stimulating a certain action. Now, there are different types of nitric oxide synthesis that exist in different types of cells. Each one of them has a certain localization, cell-specific localization, as well as different effect. But what's common between them is that all three of them require dihydrobiopterin as a cofactor, and they also require NADPH as a coenzyme for synthesis of nitric oxide. So this is the lecture for biochemistry of neurotransmitters. I want you to pay attention to differences between neuropeptides and neurotransmitters. This is one of my classic questions that I ask in the exam. I would like you to know the differences in terms of activity. So small molecule neurotransmitters act quite fast relative to the neuropeptides. Response of neurotransmitters is also in small molecule neurotransmitters is also fast, as well as but the duration of action of neuropeptides is longer than the small molecule neurotransmitters. Receptor targets for small molecule neurotransmitters, there are single receptors for each one, unique receptors for each one of them, versus receptors for the neuropeptides. So neuropeptides can bind to different types of receptors. In terms of gene expression, neuropeptides can regulate gene expression. Small molecule neurotransmitters do not do that. Note the differences in terms of synthesis, transport, and packaging of the neurotransmitters. as well as concentration. For action, neuropeptides require lower concentration for action versus higher concentration for small molecule neurotransmitter. A speed of release of small molecule neurotransmitters is different than neuropeptides. It's faster for the former and slower for neuropeptides. The concentration of calcium required for release of neurotransmitters is also different, as well as site of synthesis, modification, action, as well as finally fate. So pay attention to these differences in these categories. I know that I went fast in this lecture, but you have a chance to actually stop the recording, listen to it again, take notes. Questions will be easy, inshallah. Just pay attention to a few details here and there in terms of synthesis of neurotransmitter, and the last slide is also important as well. Again, if you have any questions or comments, you can send me an email. If you have any feedback that you would like to share it with me, feel free to be, honest, and let me know what you think. Thank you so much. Good luck. Allah ma'akum.